welcome to the DNA Lab Short Read Sequencing Workshop. I'm going to start with one of my favorite quotes from Jurassic Park. Your scientists were so preoccupied with whether or not they could that they didn't stop to think if they should. And I say this because I would like to know if you have thought about whether or not a sequencing reaction will actually test your hypothesis or answer your question. So I think this is an important thing to lead with because a lot of people sequence just because it's really neat and sort of the new thing to do. So make sure you've asked this question before you sequence because it will not be cheap, quick, or easy. It will not be the easy button that you're maybe thinking it will be. And you'll learn that this week in class. So think about it before you decide to sequence. But if you do decide to sequence, what we're going to learn in class this week is how to do the analysis. The other thing I want you to think about is what kind of sequencer you are. There's this great paper called A Field Guide to Genomics Research. It talks about the 6 different types of sequencer people, so people who sequence. And I encourage you to check it out because maybe you're sequencing for one of these reasons and that will help you to better plan out your experiments. Okay, so this is really what class is going to cover, analysis. How do you get from the sequencer down to the big P, publication? And some of you may think, although probably not, that it's like this. It is not like this. It's actually more like that. It's a big maze. There's a lot of different paths you can take, and we're here to help you figure out how to do that. So what does the real path look like? Actually, there are many paths, but this is one of them. And we're going to be going over each of these steps this week. How do you drive down these paths? The problem is that you guys have probably never driven the car you need to learn to drive. And so we will also be going over how to drive in this class. Now, some people don't prefer to think about the paths. Some people prefer to think about the stops on the path. So if you think about the stops, they're actually the file types that you're going to be dealing with this week. You're going to start with a FOSS-Q file and go through many other file types by the time you get to your publication. We're going to teach you about those. But to start, before we get into this complicated mess, this is what you will learn in this class, we have to learn to drive. So doing computational analysis of next-gen sequencing requires using Unix at this point. There are some ways you can do it without Unix, but only if your data is not large. And so we're going to be learning Unix as a first step this week. Now you're going to feel a lot like you were in driver's training class. When I was in driver's training class, I took the written exam. And let me tell you, I passed that written exam with 100% flying colors. However, then I did actual driving with a driving instructor. And I had to take the driving test in person 3 times because I hit a mailbox and forgot to turn on my blinker and ran through a stop sign. So when you're learning Unix this week, it's going to feel a lot like driver's training class. You're going to make lots and lots of mistakes. Your most common mistakes are going to be spelling and capitalization because you're not used to having to type every command. You're used to being able to click on files to open them. Now you're going to actually have to type to open files. So we're going to start this week by learning Unix. We're going to, by the end of the week, having you driving on highways. So supercomputers are like the highways for our large data. You will need to use a supercomputer if you're going to be doing anything bigger than E. coli as a model organism. So we'll teach you how to use servers and how to play nice on them. A lot of people ask me why the process of analysis is not automatic. So there are a lot of wet lab scientists out there that think that they can hand me their data, I'll push a couple of buttons, and out will pop the answer to their question they've been waiting for. I'm afraid if it were that easy, it would already be done. The thing about data most people don't realize is that your data influences your path. So even if you think you know the pathway you're going to take before you start, the data will actually change that pathway. And at every step in this process, you're going to have to look at your data to try and decide, am I going the right direction, and decide on your next direction. Bioinformatics is moving so fast at this point that your path is unlikely to be the same ever again. So even if you analyze a paper with RNA-seq data, and then in the next paper you go to analyze RNA-seq data again, you are unlikely to follow the same paths. The programs for mapping change yearly. The programs for counting change yearly. So your programs are changing, your data is changing. What that means is it's not really easy to make an automated pipeline. You can do it. It may also not be very wise, because if you make an automated pipeline, it does not necessarily taking the correct path or the best path for your data. It's just taking an okay path. So this week we're going to talk about what paths you could take. We're going to teach you one of them, but not necessarily all of them. So we start the class by learning how to drive with Unix and supercomputers. All of next week is learning the different paths that you can take for RNA-seq, ChIP-seq, and ATAC-seq. And even if you're only going to do one of those, I encourage you to come to the other days, because what it'll teach you is what different paths look like and how to find out more. Basically, our goal is to teach you how to find and read maps for bioinformatics, because you guys can do a lot of learning on your own once you know how to drive a car. We are not planning on showing you all of bioinformatics in 2 weeks. That's crazy, not going to happen. And we are not going to give you an easy button, because in data analysis, there really are no easy buttons. The pipelines may be super quick and easy, however, they are not necessarily best for your data. So I don't believe in bioinformatics easy buttons. The class is in reverse format. That's because you guys can listen to your lectures at home anytime you want on the videos that we will be providing. Really what you need is someone in the classroom working on the activities with you. So this week we have lots of volunteers from the Dowell Lab and the Allen Lab and other labs around campus who are going to help you to figure out, how do I drive? What mistakes have I made? Did you remember your turn signal? And so that's really what class is for, physical walking through the activities so that you have someone beside you. Because you are going to make mistakes, and that's totally normal. It's okay. Just remember, spelling and capitalization matter. And with that, I will tell you welcome to the workshop, and we're looking forward to have you.